Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wondered if seeing things in your mind is just like seeing them with your eyes? Well, this fascinating study dives deep into that very question, especially when someone can't see with their eyes at all. This is a big one. Researchers looked at people who are cortically blind, meaning their brains can't process visual information from their eyes. The big question is if these people can still imagine seeing things. Some scientists thought that if you damage the part of the brain that processes vision, you'd lose the ability to imagine things too, like an old VCR tape playing in reverse. But this paper puts that idea to the test. Here is the deal. The traditional idea is that imagining something is like perception in reverse. Think of it like playing a movie backward on a VCR. So logically, if the part of your brain that sees specifically the primary visual cortex or VCI is totally destroyed, you should lose the ability to imagine things too. This is called aphantasia, the inability to form mental images. The researchers scoured through wushu cases of people who were cortically blind to see if this prediction held up. And what they found was pretty mind-blowing. They discovered that many people with severe damage to their VCE could still vividly imagine things. On the flip side, when people with impaired imagination did have brain damage, it wasn't just confined to VCE, but extended to other parts of the brain. So, what is visual mental imagery, or VMI for short? It is the subjective experience of seeing things in your head when nothing is actually there. For a long time, the dominant idea in neuroscience, championed by researchers like Koslin, was that VMI is just perception running in reverse. This means that the very same pathways and brain regions used for seeing should also be used for imagining. Specifically, they believed that imagining something required reactivating the primary visual cortex VTHEA. This theory has shaped how we think about consciousness and visual processing. Evidence for this came from studies showing that when people imagined things, their VC lit up on brain scans like fMRI and PET, and zapping VC with magnetic pulses seemed to mess with their ability to imagine. But as this paper points out, this core prediction that damaging VC should abolish VMI has rarely been directly tested against what we see in actual people with brain injuries. The researchers here decided to do just that. They wanted to see if the clinical evidence gathered over a century actually supports this idea. The central prediction they tested was that bilateral destruction of VC should lead to a complete loss of visual mental imagery, or neurological aphantasia. How did they do this? They did a big literature search, looking through thousands of scientific papers from all over the world, in different languages, going back to 1888 and up to May 2024. They were looking for cases of cortical blindness, which is when someone loses vision due to damage in the primary visual cortex. But their eyes themselves are fine. They focused on cases where VMI abilities were specifically mentioned. They collected information on 55 such cases. For each case, they noted the cause of the blindness, where the brain damage was any associated symptoms like Anton syndrome, which is when someone denies being blind, and most importantly, what was happening with their visual mental imagery. They then carefully analyzed this data to see if there was a pattern. Now, for the really interesting part, the cases where people could still imagine things despite being cortically blind. Take the very first case reported by Anton in Itzenba. This patient had severe damage to her occipital lobes. The parts of the brain at the back that handle vision, her optic radiations which carry signals to VI, were interrupted. She could still vividly imagine her home and pets and even draw figures in the air. Then there is Ingem's Isinjobaisishu case. This man had complete cortical blindness due to bilateral destruction of his calcarine cortex, which is where VC is located. But he retained vivid topographical imagery, meaning he could accurately describe street layouts in San Francisco. Another powerful example is Terbrak Schenk and Van Vliet's Isenjobaisishu case. This patient had anatomically verified near-Zen total loss of VC, but produced incredibly detailed verbal descriptions of flower colors, his city hall, and clock layouts. The paper lists Shesem S. cases individually, and many more are mentioned historically. Many of these individuals could describe complex scenes, objects, and colors in great detail, even though they couldn't see anything at all. Case Shar. Bridge and colleagues Leoncelin Shar is particularly striking. This patient had a lesion almost entirely confined to the gray matter of the calcarine sulcus, which is VC yet. They retained vivid visual mental imagery. What is more, while imagining things? Their brain activity scans showed normal patterns in areas outside of VC, like the occipitotemporal, parietal, and frontal regions, just like healthy people. This case is highlighted because it is one of the closest to supporting the opposite of the original prediction. Now, let's look at the other side of the coin. What about the people with cortical blindness who did have impaired visual mental imagery? The crucial point here is to see if imagery loss occurs when the damage is confined just to VC. And the paper's finding is consistent in every published case with detailed anatomy. The lesion extended well beyond VC, for example, case C, Brockman and Van Hagen, 1996, involved bilateral occipital damage along with extensive damage to the left temporal lobe, and they lost the ability to revisualize objects and colors. Case Arshba by Chatterjee and Southwood 1996 is particularly interesting because it shows a double dissociation within the same paper. One patient with bilateral occipital damage but sparing of the left temporal lobe had preserved VMI. Another patient from the same study whose damage extended into the left temporal lobe had impaired VMI. This is a powerful piece of evidence suggesting that damage outside of V, specifically in the left temporal lobe, is more strongly associated with imagery deficits. All in all, the paper found CENSIA-R cases classified as impaired, mixed, or uncertain regarding VMI. But crucially, none of these impaired cases with detailed anatomical evidence showed damage confined only to VC. So, let's put it all together. Out of the Wushu cortically blind patients reviewed, a significant % of them, that is by 50%, had preserved vivid visual mental imagery. These are the cases that are most revealing because they show that VC can be completely destroyed or functionally lost. And yet imagery remains intact. On the other hand, for the Senchar-Pascatians classified as having impaired VMI, the critical finding is that in every case with good anatomical evidence, the brain damage extended beyond VC, typically into the ventral temporal and parietal cortex, areas associated with higher Zan-level processing. This is where the authors really drive home their conclusion. Their primary takeaway is clear and robust. Vivid visual mental imagery can indeed be spared, even with complete or near-Zan complete destruction, disconnection, or functional loss of VT. They point to several cases like Terbrak 1971, Mahar 1991, Zago 2010, Bridge 2012, and D. Jauder 2015. Which have strong anatomical or functional evidence of near-Zan total VIT loss, yet all show preserved or even normal imagery. Conversely, they found no cases of impaired imagery with informative anatomical evidence where the damage was limited only to VC. Instead, these impaired cases showed damage extending to other brain regions, most notably the left temporo-Zan occipital cortex. This finding strongly challenges the dominant perception in reverse model, which posits an obligatory role for VC in VMI. If VC were truly necessary, severe VC destruction should logically lead to absent or significantly impoverished imagery, especially when detail is required. But the cases simply don't show this pattern. The authors then address a potential counter-Zan argument. What if VV damage isn't always complete, and those small remaining islands of VC are enough to support imagery? They argue that this explanation isn't very convincing. If residual VV tissue were responsible, we would expect imagery to be restricted or spatially biased according to which part of the visual field that spared tissue represents. But they cite the Bridge et al. 2012 case again, where the patient had vivid imagery spanning the whole visual field, not just a tiny peripheral sliver that might correspond to a spared VC area. They also mentioned that historical cases with autopsy Zan verified near Zan total VC loss also showed preserved imagery. Furthermore, neuroimaging studies in healthy individuals often show imagery Zan-related activity in higher Zan-level visual areas, like the fusiform imagery node in the ventral temporal cortex rather than in VC. This provides convergent evidence that VC might not be the key player. The paper also touches on TMS studies, like the one by Koslin et al. 1999, which showed that disrupting VC with transcranial magnetic stimulation could affect imagery. While this shows VC is connected to imagery networks, the authors argue it doesn't prove VC is necessary. Stimulating a connected area can disrupt downstream processes even if that area isn't essential. Importantly, they point out that lesion evidence collected after brain injury doesn't typically have the same limitations as TMS or fMRI studies, especially when looking at cases where imagery is preserved after severe VC damage. It is highly unlikely that someone with a destroyed VC had such amazing premorbid imagery that it just happened to look normal after the injury. Another interesting point is Anton syndrome, where people deny being blind. The authors suggest that in some cases preserved VMI might actually help sustain Anton syndrome. If you can't see, but you can vividly imagine things, those imagined images might be mistaken for real perception. This is a really intriguing idea about how our brains interpret internal versus external visual information. The paper also brings in evidence from direct brain stimulation in patients. They mention historical work by Penfield and Perrotte, and more recent studies showing that stimulating temporal lobe areas can evoke complex visual experiences like scenes or landscapes, while stimulating occipital areas tends to produce only simple flashes or colors. This fits with the idea that richer, more integrated visual imagery involves higher-level brain regions. So, what is the final conclusion? The clinical record of cortical blindness, spanning over a century and wushu cases, strongly argues against the idea that the primary visual cortex, VE, is necessary for visual mental imagery. Vivid imagery is repeatedly preserved even after severe VC damage. Conversely, impaired imagery in anatomically informative cases is associated with damage outside of VC, particularly in the temporal and occipitotemporal cortex. These findings align with a network Zen-based view of vision and imagery, where higher Zen-level ventral temporal and frontoparietal systems interact dynamically. The direct contribution of this review is to convincingly show that VC is not a required component for conscious visual imagery. This figure really helps visualize the core argument. On one side, you have bilateral VC damage leading to cortical blindness. But crucially, imagery can be spared if other visual networks are intact. In the center, it shows that impaired perception due to VC damage can exist alongside preserved imagery. Then, on the other side, it illustrates the opposite dissociation. Neurological aphantasia can happen without VF damage, suggesting that problems in those higher Zen-level ventral, temporal, and frontoparietal systems are critical for imagery. Together, these patterns point away from VF being the sole gatekeeper for visual imagery. The authors also offer some excellent advice for future researchers studying imagery in people with cortical blindness. They stress the importance of using high-resolution imaging to accurately map lesions, employing thorough imagery batteries that test different types of imagery, and systematically assessing other neurological deficits that might interfere with assessments. Combining fMRI with behavioral tests and being cautious with subjective reports of vividness will lead to more robust findings. So, what does this mean for us? It suggests our ability to imagine is much more flexible and resilient than we once thought. It is not just a simple rewind of perception. Instead, it seems to rely on a complex network of brain areas, with higher-level visual regions playing a starring role. This research opens up exciting new avenues for understanding the mind's eye and how it works, even when the actual eyes can't see. It really pushes back against the idea that we need our primary visual cortex to conjure up mental pictures. What are your thoughts on this? Can you imagine vividly? Let me know in the comments below. If you found this breakdown helpful, please give it a thumbs up, subscribe to RUBIN'S Mind Lab for more science explained simply, and hit that notification bell so you don't miss our next deep dive. Thanks for watching, and I'll see you in the next one.